I think a lot of people are joining in. So I think let's start the webinar. Hi, everyone. Thank you for joining this action-packed session with Advinus, Eurofin Advinus team. Hi, my name is Himanshu. I'm from PharmaShots. We are organizing this webinar in partnership with Eurofin Advinus. And with us, there are so many people who are joining us, and it is an exciting webinar. I'm pretty sure about this. And so the topic of the webinar is successful drug discovery, collaboration among multiple functions. Just a quick note, we will be taking questions at the end of the session. So please put in your questions in the chat. The questions will be first come, first served basis. So please be very prompt during the session. You can put in the questions in the chat. So without further ado, I will make an introduction with the team who is going to be presenting. So with us, we have Berry. Berry is the Director for Business Development for Discovery Services with Eurofin Advinus team. Berry himself carries with 20 plus years of experience in business development. So Berry, over to you.
Good day, everyone. I'm Barry Feigelman. I'm your Director of Drug Discovery Services at Eurofins Advinus. For those of you who don't know us, we are the drug discovery and development arm of Eurofins Scientific, whom you've probably heard of. We're an integrated drug discovery CRO. We provide biology services, including in vitro and in vivo DMPK and tox, as well as full medicinal chemistry services. My experience includes over 20 years working with MedChem and DMPK CROs, helping scientists just like yourselves with a wide range of drug discovery and development projects. So I'd like to now take the opportunity to introduce our distinguished speaker, Dr. Punit Marathe. Punit has had a very long and distinguished career in the pharmaceutical industry, which has included positions at pharmaceutical companies, drug discovery CROs, and academia. For much of her career, she worked in the DMPK department at Bristol Myers Squibb, most recently as the head of the department. She also served as executive director of integrated DMPK and discovery services with Syngene, an India-based CRO. Dr. Marathe also holds a position as an affiliate professor at the Department of Pharmaceutics at the University of Washington. Dr. Marathe has published over 100 publications and has presented talks at multiple national and international conferences. Her areas of greatest expertise include ADME profiling of lead candidates, establishing PK-PD relationships in animal disease models, projecting human pharmacokinetic profile and efficacious doses, guiding selection of doses for FIH studies, and understanding drug-drug interactions liabilities. Punit currently serves as the VP of DMPK clinical pharmacology as well as toxicology at Agios Pharmaceuticals. In addition to her other duties, Punit also currently serves as a key opinion leader and consultant for Eurofins Advinus. So at this point, I'll turn it over to Punit. And thank you.
Thank you, Barry. And thanks to everyone who's joining the webinar today. My job is to explain to you that successful drug discovery requires collaboration among multiple disciplines. So this slide briefly summarizes the drug development process, highly simplified process in 5 steps. It starts with discovering new drug via screening and optimization of hundreds of molecules. This is then followed by preclinical research to ensure basic questions about safety. Once the molecule shows adequate safety profile in 2 preclinical species, the candidate moves on to clinical research, where the goal is to ensure that the new medicinal entity is safe and effective. It is then followed by filing of all the various reports from all preclinical as well as clinical studies to the regulatory agencies, and then the regulatory review happens of all the submitted data. And if that is found to be adequate, then the drug gets approval. Even after the drug is approved and it's on the market, drug development process doesn't end, because there are a number of post-marketing safety monitoring efforts that have to be ongoing in order to continue to ensure that the drug is safe. So the drug discovery process then is the first step of this complex drug development paradigm. And just as a teaser, I wanted to put these bullets out there to say, what is drug discovery? Is it an art of discovering a new molecule entity for patient benefit? Is it a complex process involving collaboration across multiple disciplines? Is it a highly innovative exercise that leverages cutting-edge technology driven by data interpretation and integration across functions, with an end goal to identify a candidate for entry into the clinic that is sufficiently de-risked to assess proof of concept? So to me, drug discovery is all of these, and the end goal of the process is to identify a candidate, and this to me is very important, for entry into the clinic. Not necessarily to get it approved, but the first step is to make sure that it is sufficiently de-risked and it has the ability to assess the proof of concept. And I can elaborate this further in one of my case studies. So as I mentioned, it is a collaboration among multiple functions, and some of them I have listed here. Medicinal chemistry is probably the beginning of any drug discovery that happens, working hand in hand with the biology group. And then there are all these other functions that are key support functions for the drug discovery process to succeed. And these departments include, perhaps not an exhaustive list, but based on the examples that I'm going to provide you today, DMPK, pharmaceutics, safety assessment or toxicology, translational science, and I've also added clinical research here, because without their input, it is difficult to establish a target product profile, which is very important for the drug discovery scientists to understand what kind of molecule, what kind of properties do we need to incorporate into the molecules that we are going to advance to the clinic. So these are the different stages of drug discovery, and I have probably given you one definition of how these different steps are called. Maybe they're called differently in different companies, but in my mind, it starts with the target selection, and then you have the effort to identify some hits that are hitting the target, modulating the target, depending on which mechanism you're pursuing, either an antagonist or an agonist or a reversed agonist and so on. Once we have identified some hits, this is then proceeded by lead optimization, meaning you're trying to generate several molecules and look at how they can be further optimized. An optimized lead is then selected. That's the process of lead selection. And once that lead molecule is selected, it is further characterized so as it is sufficiently de-risked, as I said, for entry into the clinic. Now, drug discovery starts with the target. So first thing to know is what target, and this is a biological target. The way that target is defined here, it's a biological entity, maybe a protein or a gene, that interacts with the new molecular entity. The target has to be relevant to the disease phenotype and amenable to therapeutic modulation. In simple terms, it has to be druggable. Now, what are the properties of a promising drug target? It needs to have a confirmed role in the pathology of the disease. It should be expressed in sufficient concentrations in select organs and tissues that is modulated, and that target that is modulated by the disease. Again, having expression in select organs and tissues is important for selectivity of the target. As you modulate it, it should be limited to the disease modulation that is of interest and not have other effects that are modulated by the target modulation. An ideal target should have a crystal structure available so that the druggability can be assessed. And we need a biochemical assay very early on that can then enable high throughput screening. There are numerous approaches for target identification validation. Some of them are listed here: crystallography, computational modeling, genomics, etc. And again, selecting the right target that is relevant for that disease is critical to improve probability of success. I'm going to showcase this with an example of two targets. One is the thrombin target and the other is factor 10A. And these are both involved in the coagulation cascade. So cardiovascular disease continues to remain a major cause of death worldwide. And even today, there is a strong need for a new antithrombotic therapies that are more effective and provide better safety. Here I have tried to illustrate two different targets in the coagulation cascade. The first is the thrombin. This is the last serine protease in the blood coagulation cascade, and it is responsible for the physiological fibrin clot formation and platelet activation. Argatroban is a direct thrombin inhibitor, which is approved for prophylaxis and treatment of thrombosis. DPC-423, shown here, is an inhibitor of factor 10A, which is a serine protease that lies upstream in the coagulation cascade, just above the thrombin, and it is intimately involved in the generation of thrombin. Now, what is thought is that rather than inhibiting thrombin, inhibiting 10A provides some physiological concentrations of thrombin to remain and to exhibit the physiological hemostatic function. So direct thrombin inhibitor, which is approved upstream of the thrombin pathway, it is thought not to affect the existing levels of thrombin and therefore not completely suppress the production of thrombin. Direct suppression of thrombin, what has led to is, as you can see here, the left side shows in the red, first let's look at the black. That is the desirable effect or percent reduction of thrombin weight. That's the efficacy against the concentration of ergotraban. The red curve here describes the percent increase in bleeding time. So this is the side effect again as the concentration increase. And as you can see, direct inhibition of thrombin, there is basically no separation between the desirable effect and the undesirable effect. On the other hand, the right side is the same curves plotted for DPC-423, which is a factor 10A inhibitor. The black curve shows the efficacy profile, percent reduction of thrombin weight relative to the concentrations. The red curve is the bleeding profile, the side effect profile, and there is significant separation between the two. So just from this, it appears that 10A is a better target for the anticoagulant effect than a direct inhibitor of thrombin. So now, once 10A was identified as the target, this led to significant SAR, and SN429 was identified, which was a very potent 10A inhibitor. However, it doesn't appear to be, and there is some selectivity over thrombin, which is 2A, but the main problem with this molecule was that it had very poor bioavailability. So only 4% bioavailability was observed in the dog. However, with its potency and the selectivity profile, it was a very good preclinical tool compound. So modification of the structure led to this DPC-423, which is what I shown on the earlier slide. This is now orally bioavailable. It has 57% bioavailability in the dog as well as adequate human half-life. However, it was discontinued in the clinic due to some toxicity. Rizaxaban was the second clinical candidate again in the same series. It also had good potency, adequate selectivity, good oral bioavailability in the dog, and adequate half-life, again discontinued due to some toxicity reasons. Now, based on the toxicity profile seen for these two agents, there was some thought about how can we avoid this potential off-target toxicity. One thought was because 10A is the target exists in the vascular compartment, then perhaps the drug needs to be restricted to the vascular compartment. If you can avoid its non-specific distribution to the extravascular compartment and therefore going into all kinds of other cells, that might be leading to this toxicity. Now, the one way to restrict vascular compartment distribution is by reducing the volume of distribution by the modification of the structure. Now, this is easier said than done, because low volume of distribution then also requires lowering the clearance significantly further in order to obtain an adequate half-life for ease of dosing, ease of frequency of dosing. So this is exactly what the scientists at Bristol-Myers Squibb achieved. And what they thought was, by changing this P4 substituent from a basic moiety to a neutral moiety, actually, they saw an observation of reducing the volume of distribution. So as you can change this P4 moiety, as you can see that it is changing, you change it here, you will remain, you maintain the potency against Na, but you are starting to reduce the volume of distribution as well. And the cyclic lactam, which is shown here as the last molecule in the table, this has the best profile in terms of the lowest volume of distribution as well as the lowest clearance in the dog. So simultaneous ADME studies here led to this molecule, which was then taken to the clinic. This is apixaban for further clinical testing. And here I am showing the profile of apixaban, which is an orally active, selective, direct reversible inhibitor of coagulation 10A. Exquisite potency, picomolar potency, selectivity against all these other targets which are in the coagulation cascade. And now looking at the ADME profile, it's got adequate permeability, actually very decent solubility, moderate plasma protein binding. This is the in vitro efficacy readout here, human prothrombin time. It is stable in human liver microsomes, very selective against CYPs as well as hERG, doesn't show cytotoxicity and doesn't show GSH adducts. So this molecule is not only on the market, an extremely useful drug for patients. This is Eliquis, and you can see how multiple collaboration among multiple disciplines here led to the discovery of this molecule. Just going one step further, again, it's a profile, selective profile relative to the bleeding risk that I was describing earlier. Here, this is being compared against warfarin, which is a very widely used anticoagulant agent. And the studies shown here are in the rabbit model. So the top graphs here show the efficacy profile. Warfarin is given orally. Apixaban in this model, rabbit model, is given IV because it doesn't have adequate oral bioavailability in that species. So this first graph here is the vehicle profile. Thrombus weight reduction is comparable between the two. And then as the dose increases, you can see the thrombus weight goes down, and the maximum efficacious dose here is 3 milligram per kilogram orally for warfarin, 1 milligram per kilogram per hour IV for apixaban, and at that maximally efficacious dose, there is about 83% reduction of thrombus weight. So both are showing good efficacy. However, now if you look at the bleeding risk, as you increase the dose here, you significantly increase the bleeding risk. The bleeding time has increased nearly 6-fold over vehicle with warfarin at the efficacious dose. Whereas for apixaban, there is hardly any increase in bleeding time in this rabbit model. Now, the point here is this animal model has been extremely useful in showing this separation between efficacy and side effects or toxicity. Can it be translated directly to patients? Perhaps not, because it's a provoked bleeding model, unlike spontaneous bleeding that you see in patients. The patients are also on polytherapy. So perhaps you can't say that, okay, you're going to have a 6-fold increase in bleeding time for warfarin, no increase, or maybe a 10% increase. Direct translation to the clinic is what is sought after. But models of this nature are extremely useful in generating hypotheses and also for retrospective mechanistic studies and exploring whatever clinical observations there are to get some insight into those, these models become extremely useful. Not to mention, they're also very useful in the rank ordering compounds when you have multiple leads to choose from. Okay, and this one now shows the same kind of profile, bleeding time, which is the toxicity on the y-axis, versus the efficacy profile, venous thrombosis percent inhibition on the x-axis, for 4 different marketed agents. Warfarin is probably the worst. For 80% inhibition of venous thrombosis, there is a 6-fold increase in bleeding time. Dabigatran, which is a thrombin inhibitor, shows again about 4 to 5-fold increase in bleeding time. Rivaroxaban, another 10A inhibitor, is comparable to apixaban, but this model shows that apixaban is better in terms of the bleeding versus efficacy profile differentiation compared to rivaroxaban. Okay, so that was the first example. Now let me switch gears and show you one other case study, and this is now focusing on medicinal chemistry efforts as they contribute to drug discovery. Medicinal chemists play a crucial role through selection and synthesis of compounds to establish structure-activity relationships. This is what is needed to enable proof of concept in preclinical animal testing with adequate safety. They use multiple tools, combinatorial chemistry, high-throughput screening, molecularly defined targets for allowing the structure-based design. And they work hand in hand with ADME scientists, safety scientists. So in vitro ADME toxicity screens as well as rapid in vivo testing, along with predictive in vivo models, this has now led to significant reduction in the cycle time for medicinal chemists for advancing drug candidates. Something about past, present, and future. So the role has significantly evolved with the advent of new technology and understanding of what is needed for successful drug discovery. So from a mix of few clinically precedented and new mechanisms, now the chemists are using predominantly new mechanisms for unmet medical need. And even today, they're using phenotypic screens and polypharmacology approaches based on translational research for target selection. Earlier, it used to be rule of 5, perhaps, for the oral drug space. Today, chemists are going beyond the rule of 5 and identifying candidates such as the PROTACs, some call them millar-molecules, peptides, etc., which are also successful drug candidates. And rather than just using in silico tools now, they're going beyond that, using better algorithms, using databases suggesting higher quality molecules for synthesis. So the role of medicinal chemist has evolved significantly, and it has become, I think, much more fruitful because of their successful collaboration with other disciplines. So I'm going to give an example of glucokinase activation. This was thought to be a very promising mechanism for type 2 diabetes. However, the issue was a significant likelihood of hypoglycemia due to activation of glucokinase enzyme in the pancreas. So if you have a systemic GK activator, it will activate the GK in liver as well as pancreas. And it was thought that the activation of this enzyme in the pancreas is what is responsible for the risk of hypoglycemia. Now, there are always clinical strategies that have been utilized to manage the risk, but a fundamental change in the molecular design perhaps was thought to be better, more effective to maximize this therapeutic opportunity in this anti-diabetic market. So thinking about a liver selective GK activator, how will this help? So this was thought that, okay, if you can restrict the molecule in the liver, a selective distribution in the liver, perhaps that will reduce the risk of hypoglycemia. However, the enzymes are similar. So structure change is not going to make them selective towards the liver GK and not bind to the pancreatic GK. So what did the medicinal chemists do to solve this problem? Their idea was to focus on liver selective tissue distribution. To do that, they had several approaches they could have undertaken. First, enable recognition by the liver-specific transporters. So make these molecules substrates for active hepatic uptake, or perhaps make them conjugates such as bile acid, so that they will be targeting the liver motif, or maybe make it prodrug, which then undergoes selective metabolic cleavage in the liver, leading to the active moiety. Now, the option 1 was chosen, that is the recognition by the liver-specific active transporters, because there is sufficient precedence for translation from the preclinical animal models to the clinic. You can imagine the prodrug cleavage could be highly, it can show big species differences when you go from rodents to non-rodents. So now if you're incorporating structural motifs that make these molecules substrates for active uptake, then it becomes imperative that along with the optimization of potency towards GK, measuring the substrate activity towards OATP has to be incorporated very early in the testing tiers. So this is an ADME assay. Perhaps it's not even a high throughput ADME assay, but it is thought to be important to be conducted early on in order to identify the suitable leads. So this is how the screening tier was then established by this research program. They first tested the biochemical activation of GK by a biochemical assay. Right up front, now you have recognition by liver-specific OATP. So is it a molecule for an uptake is it an uptake transporter substrate? Along with it, low hepatic oxidative metabolism is important, so that the molecule has a longer residence time in the liver, and therefore it is activating the target, which is now in the liver, also for a longer time. Another important property was for the molecules to have low passive permeability. And this low passive permeability now impedes the entry into other cells, such as the pancreatic cells, which do not express these uptake transporters. Now, for that, what was done was they incorporated this assay, which is a rat INS1 cell line. So this is a rat insulinoma cell line, which is measuring the pancreatic beta cell activity. So this cell potency, along with the biochemical potency, so the ratio of cell potency versus the biochemical potency was used as a surrogate measure of pancreatic impairment, pancreatic GK activation. So the bigger this ratio, that means the molecule is not as active against the pancreatic cells as it is against the enzyme. Okay, so that means it is not going into the cells where there are no uptake transporters. Very clever strategy indeed. So this molecule 17 is GK active, but it is not stable. So you can see it is potent, but it is not showing other properties that were desirable. So high intrinsic clearance, permeability is quite high. So obviously it is not going to show liver specific distribution. Its solubility is okay. INS1 potency is 150 nanomolar. Cell to enzyme ratio, which is the measure of selectivity as I was describing, is only 1.3, and it is not an OATP substrate. Molecule 19, which was the lead from this program, not only is potent, but it is showing all the other desirable properties. Most importantly, it is a substrate for OATP 1B1. It is metabolically stable. It does not have adequate permeability. Because of that, you can see the INS1 potency is rather weak, and the cell to enzyme ratio is now 77. That shows this molecule is less likely to activate the pancreatic enzyme and more likely to be selective towards the liver GK enzyme. This molecule also had better solubility. So this is now achieved by the replacement of the methyl group by the carboxylic acid group. This table shows some more structure modifications than the chemists did. So they first tried to change the position of the carboxylic acid on the heterocycle here. led to poor GK activity. Some other attempts, such as increasing the carbon length here or replacing the carboxylic acid by sulfonic acid or a phosphonic acid, led to the same result, weak potency. If you have a tetrazole, it was a substrate. Same here, oxanilic acid was a substrate, but these two molecules don't have low clearance. So in the end, this molecule 19 happened to be the best lead molecule among this series of molecules that were synthesized and went through a very elegant testing scheme to make a GK selective liver, a GK selective molecule. All right, so that was a case study emphasizing the role of medicinal chemists, although highly in a very highly collaborative involvement with other functions, most importantly, I would say the DMPK folks. So now for successful drug discovery, what we now need is the third pillar. And I call the third pillar candidate optimization. This is actually three different functions, three different disciplines, DMPK, pharmaceutics, and safety. And all these are important in drug discovery in order to identify a good molecule for the clinical advancement. And I've just listed what these different functions do in the discovery stage. So focusing on DMPK, here I have sort of differentiated what the DMPK scientists do in different stages of drug discovery. They ideally should be tailoring their studies depending on the program need. But typically during the lead optimization phase, the focus is on in vitro ADME studies, soft spot identification, if the molecule is metabolically unstable, what portion of the molecule is unstable, so that could be modified by medicinal chemistry efforts, and rodent PK to understand some early PK properties, oral bioavailability, etc. During lead selection, then you can move on to higher species PK. So you have increased confidence that the pharmacokinetic profile is adequate, not just in rodents. Here you begin to develop PK-PD relationships and further characterize the metabolic profile. Once the lead is selected, now that is further characterized by identifying the major metabolic and clearance pathways, thorough assessment of drug-drug interaction liabilities, and human pharmacokinetic and kinetics and dose prediction. So this is an overall role of ADME scientists tailored to the needs of the program depending on the stage of discovery. Pharmaceutics is also very vital for candidate success because it is actually now ensuring that you have the right developability characteristics in the molecule. So during lead optimization, the focus could be on calculated physicochemical properties and making formulations for rodent PK/PD studies. This is important because the formulation that you make, first of all, shouldn't affect, it should be safe. That's the first rule, of course, but it should not affect the efficacy readout. So you should get the right readout from these efficacy studies, and for that, formulating it is correctly is quite critical. During lead selection, the effort now goes into measuring the physicochemical properties and formulation for definitive PK/PD studies. And then in lead characterization, there is a assessment of solubility, stability, selecting the material and formulation for tox studies. Important again, because once the formulation is identified for these tox studies, it is in our best interest not to change the formulation because that can lead to change in exposure and that will then require bridging toxicity studies. Safety assessment can actually make or break the molecule. So early on, important for them to assess target expression in various tissues. If the target is expressed in various tissues, it is then likely to have off effects that are beyond the desirable effects. On target pharmacology, and it is assessed by utilizing knockout, knock-in animals to get some understanding about what that target modulation is likely to lead to. During lead selection, there is focus on off-target effects, significant in vitro investigative studies, depending upon what the observations are in maybe efficacy models. If you start to see some toxicity, maybe try to understand where it is coming from. Is it species specific? Is there a role of a metabolite, for example, and so on? Is it chemotype based? So if you change the chemotype, can you get rid of the toxicity? Safety assessment has to work hand in hand with the rest of the discovery team for this. And then during lead characterization, there is gene toxicity, acute and repeat dose toxicity, and safety pharmacology studies. All these are before the definitive GLP compliant IND tox studies are done. So this is all that the safety scientist is offering during the drug discovery phase, again with the goal of sufficiently de-risking this molecule so that it could be advanced to the clinic for proof of concept. Okay, I'm going to switch to another case study here, and this is now focusing on the DMPK efforts. So example is of SGX-523, which is a MET inhibitor for the treatment of solid tumors. It demonstrated species-dependent toxicity. It actually went into the clinic and patients exhibited compromised renal function, presumably resulting from crystal deposits in renal tubules. Why was it not discovered in the preclinical testing? When further metabolite profiling was done, that revealed a major NADPH independent late eluting metabolite, which was generated by monkey and human liver S9, but to a lesser extent by rat S9, and it was absent in the dog S9. So this was the reason. It was not present in the dog S9, and it was present but only to a small extent in the rat S9. Rat and dog being the tox species, this toxicity was not observed in the preclinical tox studies. So further efforts were undertaken in now liver S9. So if the efforts are concentrated in liver microsomes, which is usually the case, what happens, this typically then downplays the role of non-P450 enzymes. One such enzyme is aldehyde oxidase. It is a cytosolic enzyme. So if you do the studies only in liver microsomes, you are not likely to understand any contribution due to this enzyme. Now it is complicated, this enzyme, because the human activity is relatively high compared to the commonly used tox species. It is there, but in a smaller extent in the rat, it is absent in the dog. Monkey is the species that comes closest to human in terms of aldehyde oxidase expression. So here you see the liver S9 profile in the rat and the dog and the monkey and human. You see the monkey is closest to human. This metabolite here is this 3-quinolinone metabolite, late eluting, and this has significantly less solubility compared to the parent. Inhibitor studies also confirmed the role of AO. And some mechanistic studies also confirm the role of AO because the oxygen was incorporated from water instead of instead of the atmospheric oxygen. When the studies were done in monkeys, this resulted in the metabolite. Metabolite was excreted in the urine far greater than the parent. So the key message here is choosing the right species for the tox studies is critical. And this was done by, this could have been avoided if the metabolic profile was assessed thoroughly and therefore leading to the selection of monkey instead of the dog as the non-rodent tox species. Okay, now the last case study is focusing on the translational research. The example I have taken is published by some scientists at Pfizer. It's a relatively old paper now, 10 years old, but is such a classic paper in this area, and it highlights the importance of translational research so vividly that I always like to take this example. These scientists described 3 pillars necessary for success. Now, they defined success as the ability to go from phase 2 to phase 3, meaning you have achieved proof of concept in phase 2. So this is the, again, key message I come back to, that successful drug discovery is to be able to identify a candidate that has the ability to test that proof of concept in the clinic. meaning it has to be sufficiently safe and it has to reach adequate exposure such that we can test the mechanism of action, the target modulation. The three pillars are as follows. The pillar 1 is the target exposure. Is the exposure at the target site sufficient? Does it meet the desirable levels based on potency? If it is a peripheral target, plasma level is sufficient. If it is a central target, obviously, we have to make sure there is adequate brain penetration. Pillar 2 is target engagement. What if the drug gets to the target in adequate levels, but it doesn't show adequate potency? There is not sufficient target binding. So is there sufficient binding to the pharmacological target in the target tissue that meets the predefined criteria? Pillar 3 is the downstream event of target engagement, which is target pharmacodynamics. Is there data showing the desired pharmacology in the target tissue meets the predefined target area? Do we see subsequent downstream pharmacodynamic change that is a result of this target modulation? So if you satisfy all three criteria, pillar 1, 2, 3, that improves the probability of success. And that's shown here by this plot here. There are 44 assets here that went to phase 2. Now, the scientists have shown this as exposure confidence on y-axis versus pharmacology confidence on the x-axis. You will note this bottom lower right block where there is plasma exposure, but there is no target binding and poor understanding of PK-PD relationship. When that happened, actually, this was the reason they identified for no phase 3 starts. 0 out of 12 assets in this bucket led to beginning of phase 3. The left upper bracket here, this is where there were 2 out of 12 phase 3 starts. In this case, there was exposure, adequate exposure, so exposure confidence is high. In the relevant compartment, there was target binding, yet there was no PD. So pillar 1 and pillar 2 were okay, but pillar 3 criteria were not met, 2 out of 12 phase 3 starts. The lower right, there is plasma exposure and target binding again, but no PD, no downstream event, 0 of 6 phase 3 starts. However, when there was target exposure in the relevant compartment, there was adequate target binding and expected PD effects were shown. 8 out of 14 assets led to phase 3 starts. So what this means is there are actually remaining, there are still 6 assets here that did not start phase 3. So they didn't even, you don't even know what happened to those molecules that you were not able to achieve a proof of concept. I think focusing on these 3 pillars actually illustrates what is key in terms of the translational research to improve the probability of success. And then this brings me to what is translation research in some simple terms. This, you could consider this as an application of knowledge from bench to bedside, but the questions that you have to ask in the discovery phase is how are you going to quantify this effect of target modulation in vivo? And this is important because efficacy studies could be very long, very prolonged, very expensive. So we have to get an evidence, firm evidence of target modulation early on, perhaps even after a single dose, perhaps even in healthy volunteers. What can we do to assess that target modulation? When we are leveraging these animal models extensively, how is the underlying disease process in animals relate to what are you trying to treat in the clinic, in patients? What is the mechanism of action? How does that relate to? Which patients are most likely to respond to this target intervention? How are you going to select the patients in your trials? What kind of characteristics, genomic, proteomic characteristics these patients need to have? How are you going to measure the evidence of target engagement in the clinic, the downstream pharmacodynamic effect, and disease modulation? So having these right biomarkers identified early on before the molecule is advanced to the clinic is key to the success of clinical research. So this brings me to the last slide. I think I hope I have explained to you that drug discovery is indeed an art of discovering a new molecule for entry into patients, but it is for patient benefit, of course. It is a complex process that involves collaboration across multiple disciplines. It is a highly innovative exercise that the success of which depends on leveraging key technology, cutting-edge technology. It's driven by data interpretation and integration across functions with the end goal of identifying a candidate for entry into the clinic that is sufficiently de-risked to assess proof of concept. Thank you.
Thank you, Dr. Punit. It was really exciting and very informative. And kudos to you. Within 45 minutes, you have completed. That's, I think, the first time I have seen so much information and very elaborately and still beating the time. So kudos to you. I pass on the information to Barry now. So Barry, maybe we can start with the questions. We have a few questions here. So maybe Barry can start asking questions, and you can answer those questions. Barry, to you.
Okay. So yes, so at this point, we're open to questions. If you have questions, please post them in the webinar chat, and Punit or I will be happy to answer them.
So I think I'll start asking the questions. There are a couple of questions here. So someone has asked, how you see India for outsourcing drug discovery hub or destination in next 5 years, and CRO's role to support the discovery.
Barry, do you want to take this?
Please repeat the question.
Yeah, sure. How you see India for outsourcing drug discovery hub? So how do you see India as a drug discovery hub or destination in next 5 years, and the role of CROs to support that discovery?
Okay. Well, yeah, India is definitely growing rapidly as a destination for drug discovery CROs, and there are quite a number of very professional CROs currently in India, and I would expect over the next 5 years that India will become even more prominent as a source for drug discovery services. The area of Hyderabad, Genome Valley, is very rapidly growing. There's a lot of investment there, a lot of companies that are springing up there. Plus the geopolitical situation with China is making a number of companies want to diversify their vendors to include India. So yeah, I do see a bright future for Indian drug discovery development CROs.
Yeah, I can just add that there is absolutely no shortage of talent and sheer enthusiasm. I mean, whenever I visit India, I am just amazed by the energy level of the scientists, and I feel young again just by being with them. So that's I think that's a really promising future that and India can play a significant role in drug discovery.
To add to what Barry was saying about the geopolitical, I would also like to add here is that even government is making a lot of efforts to to change the policies within the CRO industry, making a lot of changes. Ease of doing business is also improving here. So I think that adds to the easeability for companies to share the work here in India. I hope we have answered this question. I'll move on to another question. Another question is anonymously asked, what all early in vitro testing methods to predict drugability issue?
So this is a very broad question. I think if you're talking about just hitting the target, you just have to maybe understand the properties of the target. And there are approaches, structure-based design, ligand-based design, that enable honing into a lead series quickly. And once that lead series is identified, I think then from then on, it becomes a lot easier. I mean, again, it's never easy. It's a complex process because you're optimizing multiple parameters here. But if you're talking about druggability in terms of hitting the target, then I would say first making sure that the target you've selected is right. And then second, you have the early leads, whichever design approach you're using is important.
I hope this answers the question. Thank you, Punit, for that. Another question for Mr. Vishal. When do you think metabolite profiling and their effects on both efficacy and safety should be done?
Okay, so I think early on, of course, we at least need to understand is the if the drug is breaking down very rapidly. So that's only a question of rate. It's not necessarily profile. But I think once the lead is selected, it is important to know how many metabolites there are. If you have a reactive metabolite, that's important. That kind of testing is important because that it goes hand in hand with safety assessment. And also if there are any active metabolites, this is important. So I would say as you are in the lead selection stage, these things are important because if there is a significant contribution from an active metabolite to the pharmacological activity that you're seeing in your efficacy models, it's important to know that that metabolite is formed in humans as well. Otherwise, you'll be significantly overestimating the pharmacological activity of the lead you've selected.
Thank you, Punit. Another question comes from Rajanpreet Singh. What is the role of project management skills in drug discovery? I know it's a pretty broader question, but try and cover the base points, please.
I mean, again, it depends on whether it's a partnership. You're working as a CRO with a client. Then obviously, project management plays a key role to just coordinate the different aspects of the project, the progress of the project, different studies, communication between different functions, the CRO and the client, and so on. But even if you have your standalone drug discovery company and it's all internal discovery, maybe it's not absolutely necessary to have a project manager, but then again, it always helps so that the scientists can focus on science.
Yeah, I would add here that having a project manager allows our clients to have a single point of contact, to basically coordinate the study, to coordinate the progress of the study, to coordinate sharing of data, and to coordinate having regular meetings to discuss the project. So a good project manager makes the project go a lot more smoothly, I would say.
Punit and Barry for the reply. Another question comes from Mr. Yogesh. What is Industry 4.0 and digital transformation's role in drug discovery? Again, it is a pretty broader question, but we can touch bases on it.
It is a very broad question. I wouldn't know where to start.
Maybe we can talk about some, newer trends probably, and yeah, Mr. Yogesh can touch base with the Eurofin Advinus team later after the call also. Okay, I think we'll take up the next question. Punit, it's for your case study. The case. Mr. Ashwin Kumar asks, what happens in the last case study where the target or enzyme is central for CNS therapy area, but modulation of the enzyme is peripheral and the effect is central?
Yeah.
In this case, CMP doesn't reach to central 0% brain penetration. How do you see these three pillars will help in such case? The translation does not fit well. And in my experience, the enzyme does modulate in CNS and deliver desired effect. These are chances to miss out on target engagement.
Sorry, I didn't quite get the question.
Okay, I'll repeat this. What happens in the last case study where target is central to CNS therapy area, but the modulation of the enzyme is peripheral and the effect is central? How do you see these three pillars will help in such a case?
I see. Okay, very interesting. So the target is peripheral, but the effect is central.
Yeah.
So this has to be somehow, perhaps the target modulation peripherally leads to a pharmacodynamic downstream change. Let's say I'm thinking something like a cytokines, for example, or some inflammatory markers that are altered now peripherally, and now they are able to cross the blood-brain barrier and modulate the effect centrally. Maybe one example I can think of. So again, the three pillars are important because now if you think the target exposure in the brain is not, the drug exposure in the brain is not necessary, that's fine. Plasma exposure, modulation of target, that's fine in periphery. But then the downstream event, we have to make sure we can demonstrate that is happening centrally. And perhaps you will understand what is that link between target modulation in the periphery, but the downstream event in the central space. If you understand that translation, and then see how maybe the animal species that you're using correlates to how you would see this in human, that could lead to greater probability of success.
Thank you. And to Mr. Ashwin, I hope this answers your question. And if in case you want to discuss it more, maybe you can again reach out to the Eurofins Advinus team and have a detailed discussion, maybe set up a call with Punit and the team and have more broader discussion on this. So we don't have any more questions, Punit. Thank you everyone for joining in, those who are still now in the call and some of you who have dropped. So thank you all for joining in. It was really an interesting session. Thank you panelists, including Barry, Puneet, Ritika and Yogesh for your presence, and the recordings will be shared on our YouTube channel, and it will be shared individually over the mail also with all the registrants. Presentation will not be shared, but maybe you can read from the from the video. The feedback form will also be circulated to all the registrants and all the attendees. Please feel free to fill the feedback and share your feedback. It will be helpful for us as well as for the Eurofins Advinus team as well. Please feel free to share your views as well, and stay tuned on PharmaShots. We'll be coming up with more such informative webinars. Thank you. Have a nice day.
Thank you.
Thank you, everyone. Have a nice day. Goodbye. Good night.
Thank you so much.
Thank you. Have a nice. Bye-bye.
Bye-bye.